Hello, my name is Joel Moncur, and I'm from the Joint Pathology Center in Silver Spring, Maryland. Welcome to our series on using proficiency testing data to improve the performance of your molecular laboratory. This talk will focus on the comparison of molecular oncology laboratory-developed tests and FDA-approved assays. These are my disclosures. Okay, the bottom line up front for this talk is that PT data demonstrate excellent performance for both laboratory-developed tests and FDA-approved assays for BRAF, EGFR, and KRAS. Both types of tests exhibit strengths and weaknesses that can help laboratories with assay selection and validation, and that most laboratories using FDA-approved assays reported modifying the approved procedure, which renders it a laboratory-developed test. So with that, I'd like to give you a little bit of background. So we all know that there's an ongoing debate about the regulation of molecular assays that's focused on analytical performance. And one of the frequent assumptions is that FDA-approved assays have superior performance compared to laboratory-developed tests. So it raises the question, are there any studies that directly compare performance? Well, proficiency testing, or PT, is an absolutely excellent way, if not the best way, to directly compare laboratory-developed tests and FDA-approved assay performance. It compares hundreds of participating laboratories, and there's a range of institution types, such as academic, non-academic, commercial, US, non-US. Their laboratories are assessed over multiple time points, and they oftentimes have a diversity of platforms and assays that they use. So the CAP Molecular Oncology Committee conducted a study that was published in 2018, comparing the performance of laboratory-developed tests and FDA-approved assays for BRAF, EGFR, and KRAS proficiency testing programs. You can see here the number of participants in each of the different PT programs. For BRAF, there are 212. For EGFR, there was 197. And for KRAS, there were 282 unique laboratories. Again, they were assessed over multiple time points with multiple samples that contained a mixture of both common and uncommon mutations. The first thing that we identified as part of the study was that there were more participant laboratories that reported results for laboratory-developed tests than for FDA-approved assays. You can see here for BRAF, EGFR, and KRAS that the number of participants using FDA-approved assays or using LDTs was anywhere from 3 times to 7 times as many than the FDA-approved assays. When looking at performance, both laboratory-developed tests and FDA-approved assays exceeded 97% accuracy on their PT assessments for BRAF, EGFR, and KRAS combined. Here you can see a bar graph showing LDTs compared to FDA-approved assays, and they're both excellent. There were, however, gene and variant-specific differences in performance between LDTs and FDA-approved assays. So for instance, for BRAF, as you can see in this table, LDTs performed significantly better than FDA-approved assays. And for EGFR, the converse was true. FDA-approved assays performed significantly better than LDTs. And for KRAS, there was no significant difference in performance between the two. So the difference for BRAF, why LDTs perform better, was because of this variant, V600K, which is a multinucleotide variant. It predicts a favorable response to BRAF inhibitors for some tumors like melanoma. And the reason that LDTs perform better is that the most commonly used FDA-approved assay was actually not designed to detect this clinically significant BRAF variant. It was approved before this variant was commonly tested, and just simply wasn't included in the test design. So for EGFR, the reason why FDA-approved the variant that was accounted for the difference between FDA-approved assay performance in LDTs was a single nucleotide variant called L861Q. This comprises 2% of EGFR mutations in lung adenocarcinomas, and it's associated with low efficacy or complete resistance to some EGFR tyrosine kinase inhibitors. And we did an exhaustive investigation to try to understand why the FDA-approved assays performed slightly better than the LDTs, and we're unable to identify an explanation for that. So we were interested in tracking the performance of these particular variants over time. So since 2015, we aggregated all the performance of labs for these 2 different variants that were included in PT programs multiple times. And both LDTs and FDA-approved assays have exhibited over 97%, equal to or greater than 90% accuracy for both BRAF and V600K and EGFR L861Q. One other interesting observation from the study is that more than 60% of laboratories using FDA-approved assays modified the approved protocol, which rendered it a laboratory-developed test and needed to be validated. Those modifications included validation of new specimen types, such as fine needle aspiration specimens, and changes to the FDA-approved lower limit of detection that labs were able to validate on their own. So back to my bottom line or conclusions, PT data demonstrate excellent performance for both LDTs and FDA-approved assays for BRAF, EGFR, and KRAS. Both types of tests exhibit strengths and weaknesses that can help laboratories with assay selection and validation. And the majority of laboratories that use FDA-approved assays report modifying that procedure, which renders it a laboratory-developed test. I'd like to point out that the CAP maintains on their website a molecular oncology topic center that contains publications from the Molecular Oncology Committee, including the publication that I've described today, as well as many others. And you'll find that at the website that's listed here. Thank you very much and have a wonderful day.